by Professor Zhu. Okay, he is going to talk about the targeted treatment of bacterial infection using silicon nanoparticles.
Right. Thank you. All right. Good afternoon, everyone. Thank you for your kind introduction. My name is Jinmyoung, came all the way from South Korea. It's my first time to participate in CLINAM meeting, and really enjoyable. And I'm so pleased to have this opportunity to share my latest research interest. So what I'm talking about today is biodegradable silicon nanoparticles for targeted delivery of bacterial infections. So as we know, in conventional point of view, the drug discovery has been focusing on finding certain chemicals or biologicals that has effectiveness to treat certain disease. But in nowadays, some nanoparticles has been contribute to or support some enhance the effectiveness of the already known drug molecules, or like by using this certain nano platform like the Toxicity. And in the future, as you guys the nanomedicine can become an active principle in the drug discovery, the field of drug discovery. So there are several nanoparticles or nanomedical platform already has been approved by the FDA and used in clinic, such as the Toxicity or Asan. Most of their platforms are based on proteins and liposome, and some iron oxide nanoparticles applied and also approved, and they used it for the MRI contrast agent. And but there are some fascinating functional nanoparticles, especially the quantum dot or semiconductor nanoparticles. They are over the decade, many research groups has been reported the potential of quantum nanomaterial as a targeted nanomedical platform. But no matter how strong it is, their use is still limited in the bench-style research. The most common concerns of the quantum dot is their toxicity, because most of the quantum dot, especially the early stage of quantum dot research, has been based on the heavy metal containing quantum materials such as the cadmium and lead. And so no matter how powerful or no matter how fancy it is, you may not like to inject the cadmium sulfide in your bloodstream. And also the FDA has requires the injected nanoparticle in the human body, especially to the diagnostic agent, must be completely cleared in a reasonable amount of time. So what I'm going to talk about today is the silicon-based nanoparticles. So this is the silicon micron particles, as well. And the silicon has the intrinsic photoluminescence in the near IR ranges, and also it has porous structure, so you can securely load certain drug molecules inside the porous structures. And this is a simple translation of silicon quantum dot on your fingertip. So you can draw down the letter on your skins with the silicon quantum dot. And as I said, the silicon has readily biodegradable in the physiological condition. Also, they are very biocompatible, and they has intrinsic photoluminescence. So you don't need to label certain fluorescence dye onto the nanoparticles. So it is luminescent by themselves. And so the silicon chemistry is very robust and precise, as reported in a bunch of papers. So what we do is starting from the silicon nanoparticles, and then we do the surface chemistry to load certain drug molecules, such as the siRNA or microRNA, as well as the small molecular drug inside the porous structure of this porous silicon nanoparticle. And then after we do the screen the targeting peptide that specifically homes the disease sites, such as tumors or infections, and we make this kind of platform. So and then the next task would be the systemic delivery of nanoparticle to the disease site, in this case, the brain injury. So no matter how you design fancy in a fancy way, most of nanoparticle injected through the bloodstream has very, very low delivery efficiency. So a bunch of nanoparticles are cleared in your immune system and also accumulate in other organs and then do some induced the side effect. And a very small bunch of nanoparticles injected through inject your bloodstream can be reached to the disease site. For example, in the penetrated brain injury model, we try to enhance or improve the targeting efficiency. We conjugated the RBG peptide, which is derived from the rabies virus glycoproteins, that has a specific interaction with the acetylcholine receptor on the neuronal cell. So once you conjugate the RBG peptide to a nanoparticle, that has a specific interaction with the neuronal cells, and then you can enhance the delivery efficiency of siRNA in vitro. And we also made the mouse model of penetrated brain injury, and then inject the peptide conjugate nanoparticle through the tailbone, can get some enhance the targeting efficiency at the injured site of the brain. Also, the siRNA, which is labeled with fluorescent dye, also can be accumulate, also target the disease site. But what we really want to do is to find certain targeting peptide that has enhanced or specific homing efficacy to the injured site. So we utilize the in vivo phase display method and then try to find a certain sequence of a peptide that specifically target the injured side of the brain. And we do find that the short peptide, the sequence that CAQK has very strong specificity or homing efficacy to the injured side of the brain in our mouse model. And this peptide labeled with the FITC shows very high accumulation of this peptide to the injured side of the brain, while the control peptide does not show any targeting efficacy. And also we found this peptide co-localized with conjugating sulfate receptors in the area of brain injury. And we eventually want to deliver the siRNA to the injured side of the brain. And then we show but at the moment we don't have any proper model of mice to show RNA interference at the injured side of the brain. So we do use the transgenic, the GFP expressing mice, and then we can show the CAQK conjugate nanoparticles successfully deliver the siRNA to the site of a brain injury, and then they show superior knockdown of GFP at the site of injury. And then if you compare the targeting efficiency with RBG and the CAQK, the RBG does not specifically target the injured side of the brain. It normally targets the neuronal cell, interact with neuronal cell, but the CAQK has superior targeting efficiency of the injured side of injured tissue or cells in the side of the brain. And more recently, we try to find a certain model to target the Staph aureus infection, the target Staph aureus. So we do make Staph aureus induced pneumonia model in mouse, and then also applying the in vivo phage display, and then find a certain peptide sequence that has specificity to the Staph aureus in our mouse model. And we found the ARG motif has a specificity to the Staph aureus. And in vitro system, the peptide can specifically bind to the surface, the membrane of the Staph aureus, and also in vivo, this is the long section of the Staph aureus infected mice, and we can see the green signal, which is from the FISC. The peptide specifically target the infected lung side, while the healthy or sham infected mice does not have any specific accumulation of this peptide. One surprising thing is the cargo peptide can recognize the MRSA as well, so which is communicable associated strain can be found in the clinic. Also, the cargo peptide also target the other strain of Streptococcus. So we do say the cargo peptide has like general targeting efficacy all of the Streptococcus gram-positive pathogens. And we also tested this same peptide to target the Pseudomonas, and then this peptide does not recognize the Pseudomonas in vivo. And also, once we compare the efficacy of the Pseudomonas targeting peptide to the Staph aureus, and this does not have any specificity. So we can say the cargopeptide has a strong affinity and a strong targeting efficiency to the Staph aureus, not the Pseudomonas. And we also find the cargopeptide co-localized with Staph aureus with inflammatory leukocyte, as also they are co-localized with the host phagocyte. So that is a very, very surprising thing. So even though the Staph aureus are, even the phagocyte cells harboring in the Staph aureus, they can be also targeted by this peptide. That means we can deliver the antibiotics to the Staph aureus that is uptaken by the phagocytic cells. So for the last stage, so we try to deliver the antibiotics, in this case of vancomycin. These vancomycin molecules can be loaded to this porous nanoparticle using by formation of the calcium silicate layer onto the surface, so we can mechanically trap the antibiotic molecules inside the pore, and then we can monitor the drug release kinetics simply tracking the luminescence signal. So once you inject this vancomycin loaded and cargopeptide conjugate nanoparticles through the tail vein to the lung-infected mouse model, you can get very high specific targeting efficiency at the site of infection of lung. And there are certain some unfavorable non-specific accumulation of the nanoparticles to the liver and kidney, but the infected lung has very much enhanced the targeting efficiency. And then once we tested the survival test, this Staph aureus induced pneumonia model has very, very acute modality, so they only has like less than 20% of survival within 7 days of the infection occurred. But once you inject the targeted nanoparticle loaded with vancomycin that has 100% survival rate, we say, but what if you inject the free vancomycin, which has the same dosage of the of the molecule that loaded to the nanoparticle only has like 30% or like 30 to 40%. So that means this targeted nanoparticle system much enhanced delivery efficiency of the vancomycin to the site of the infection. So we can get 100% like or much enhanced survival rate by using this targeted nanoparticle system. So that means we can use only 10 or like 10 for the less vancomycin doses to treat the same bacterial infected pneumonia model in our mouse model. And once we study on the histology, we can see the recovery of the infected tissues as well. So in summary, we try to screen the certain the homing peptide that specifically target the brain injury or the infected pneumonia model. So and then once you conjugate this homing peptide to a nanoparticle, which is loaded with certain therapeutic agents such as small molecular drug or oligonucleotide, you can specifically deliver this medical platform to the site of disease. So what we do is starting from the material synthesis and do surface chemistry and do the biomedical technology, and then we get some feedback in all these systematic studies, and we are trying to translate our benchtop technology to the in the translational region. So this is our international collaborator in our lab, and yeah. So thank you for your kind attention. I will wrap up my talk right here. Thank you.
So, question, please. Okay, actually, you started your the presentation using traumatic brain injury. So I was trying to follow up your study, but finally you talked about the methicillin or the vancomycin resistance step infection, how to control that. And the beauty of your study in one side is just you have developed a nonapeptide, the targeting ligand for a step. And the other side is, was it delivered? Your silica was delivered systemically to. So
yes.
it accumulated in the infection focus very well.
What do you mean?
What I mean is just a pneumonia model, and how did you administer?
Oh yeah. Also in the pneumonia model, we do inject the nanoparticle through the TheraVane or like the intravenously. So they are also systemic delivery.
So how often?
Just one time.
Just one time.
Yeah, just one time.
And with the almost 1/100th of amount of vancomycin.
Yeah, like the 10-fold less amount of the free vancomycin can be used to treat the pneumonia model in our
So
case.
very interesting, and which is just published in Nature Biomedical Engineering. And if
early this year.
anyone is interested in, then they can follow.
Thank you very much.
Thank you.
Thank you.